Welcome to the University of Wyoming Plant Sciences Seminar. I'm coordinator for the course, and I'm presenting seminar today and a webinar. I'm Groose. I breed plants and teach genetics at the University of Wyoming. A couple of weeks ago, my good colleague Dr. Kniss asked the question, what have they done to our food? I'm here today to answer the question, well, we've genetically modified it. This is somewhat prompted by a very good conference we had at the University of Wyoming earlier this month. A lot of interesting topics discussed. One of them was genetic engineering, and we only heard one side of the story. And Dr. Kniss and I are maybe blowing off a little steam, responding to this and doing something for our mental and emotional health by doing so. Pretty much everything we eat has been genetically modified one way or another, and probably in many ways. And I'm focusing on plants, okay, Department of Plant Sciences, whether it's plants for food, feed, fiber, biofuels, or flowers, all different kinds of genetic modification are utilized to improve plants. Flowers, looks lavender to me, but in the business, this genetically engineered rose is referred to as a blue rose. I'm going to do sort of a question and answer format in my presentation here, like so. Not all of my answers are definitive, or maybe not everyone would agree with all of them. And I would also say, remember, we're all entitled to our own opinions, but we're not entitled to our own facts. So, GMO. Oh my God. What is genetically modified? I'm going to take a little time with this slide. This is my definition, and it's the definition that appeared in a report by the National Academy of Sciences that we'll return to towards the end of this seminar webinar. Although in popular parlance, the term genetically modified, GM, often is used interchangeably with genetically engineered and biotechnology. I'm paraphrasing some here, and there's more than one biotechnology, as we'll see. GM refers to a range of methods used to alter the genetic composition of a plant or animal, including traditional hybridization and breeding. GE is one type of GM that involves making an intentional targeted change in a plant or animal gene sequence to affect a specific result. And usually that targeted change with genetic engineering is adding a gene from some other organism. At the time that this report came out 10 years ago, targeted, you might put quotation marks around that. It might have been somewhat at random as to exactly where the transgene inserts in the genome, although nowadays genetic engineers, the gene jockeys, actually have ways of putting genes in specific places. So, what are genetically modified plants? We've got a broad definition that I'm using. We've got that narrow definition that the press and I guess the anti-genetic engineering crowd tends to use, referring just to transgenesis. Either way, I think the desired outcome is genetic improvement. Why genetically improve plants? Well, a somewhat busy slide that we use in plant breeding class. Plants are a product of their genes and their environment. Okay, and sometimes those genes can modify how plants interact with their environment, physical, biotic, etc. Ultimately, I think our goal is What I'm saying is sustainable and sustainable agriculture in terms of both the environment and profitability for growers, among other things. Why genetically improved plants? Well, we've got more mouths to feed. We're reasonably successful at that. There is a demand for more meat around the world. I don't think it's going to go away. Some of us, maybe myself included, probably could get by consuming a little less meat. But why genetically improved plants? We're actually doing pretty well in some respects. Population's going up, and fortunately at the same time it's taking less land per person to feed people. And we'd like to be able to set aside some land for other uses, enviros like me, like wilderness. Why genetically improved plants? Look at wheat yields in developing countries. Food per person is up worldwide. Yes, many people go to bed hungry, but on the whole, worldwide, people are better fed than ever before. And some of us are probably overfed and maybe to some extent undernourished at the same time. But, gee, those are issues that maybe go beyond what we're discussing today. Do we need another green revolution? There's one of my heroes, Norman Borlaug. He worked with corn and wheat. He won a Nobel Peace Prize. I think he's the only plant breeder, actually, who was a breeder producing cultivars to win a Nobel Prize. And it was the Peace Prize. And I guess the rationale there was that if everybody's got enough to eat, the less likely they are to fight. By the way, which green revolution rice would you rather grow? The one on the left or the one on, or which rice would you rather grow? The one on the left or the green revolution on the right? Often students will immediately pick the bigger, taller plant, but that one on the right is actually putting more biomass into grain. These semi-dwarf varieties are so important in rice and wheat. The improvement in corn, I think, mostly has to do with denser populations and some physiology. By the way, something that's often overlooked is that we're often not working with individual plants, but with populations of plants. I don't have time to give you a course in either quantitative genetics or population genetics here, but all of these evolutionary forces that can change frequencies of genes and genotypes operate in the wild and are utilized by plant breeders in improving crops. How do we describe selection in nature and in agriculture? Again, not time for a thorough discussion of quantitative and population genetics, but we've got equations and everything. A derivative of Fisher's fundamental theorem of natural selection, basically Fisher put Darwin into mathematical terms, derivative is the breeder's equation, describing artificial selection. And GS sub S is our gain from selection for a particular trait. There are several components here regarding our intensity of selection, variance that we've got, heritability of traits. We can manipulate those parameters in plant breeding. So natural versus artificial. These terms go back to Darwin, and we'll work bottom up here. Of course, Darwin, ideas of evolution had been around a long time before Darwin, but it was he who discerned the mechanism of natural selection. My example up here, by the way, is maize, which started out as teosinte with a hard carapace around each kernel. Eventually, at some point, there were some mutations that put kernels on the exterior of a cob. So we're starting out with heritable variation in fitness driving evolution by natural selection. Well, it was hunter-gatherers who selected favorable plants. They didn't have pre-designed breeding programs. And by the way, most of this work, the domestication of maize, domestication of wheat and barley in the Fertile Crescent was done by some hard-working Neolithic women while the boys got to go hunting. Well, and they contributed some high protein to the diet. Artificial selection. You know, Darwin was actually, besides his trip around the world on the Beagle, he was very much impressed by the gains in the improvements in plants and animals that agriculturists had produced. And in fact, the first chapter, as I recall, of Origin of Species has to do with agriculture. He referred to the selection that agriculturalists were doing in those days and now as artificial selection. He wasn't implying that what we do is something fake, like artificial flowers. He meant artificial in terms of producing an artifact, like those Neolithic people were making stone axes, making spears, well, domesticated plants being improved as new varieties can be considered an artifact. How are cultivated plants genetically improved? Well, breeding and now biotechnology, and the two really complement one another. On the breeding side up there, I've got hybridization and selection. That's pretty much the core of modern plant breeding, at any rate. The sciences upon which breeding is based are classical genetics and statistics. Biotechnology, I'd break biotechnology actually out into three different areas. There's all of the cell culture and micropropagation technologies, in vitro technologies, there's many. Molecular markers, kind of quiet, doesn't get a lot of press, but being used big time by the big seed companies. Going to the genome, sequencing markers to find aids to identifying chromosome segments that contribute to important traits. Cell biology and molecular genetics would be the basis here. Oh, I left out genetic engineering. That's the one that does get all the press. We'll have something to say about that. Transferring genes from one organism to another using recombinant DNA technology. The goals are the same as far as I'm concerned, which is genetically superior plants. What kinds of cultivars are genetically improved? All kinds. Breeders break plants out into four fundamental populations in terms of the breeding that is done. We have plants that are bred as inbred purelines, open pollinated populations, hybrids, and clones. Here are some schematics illustrating what it is breeders do. This is actually at the core core of the plant breeding course that I teach. I previewed this with my plant breeding students on Wednesday, asked them what I could cut out that might not be necessary. They told me this. This is the core of the course. Anyway, and now aided by myriad advanced breeding and biotechnology techniques. As far as Wyoming examples go, wheat, barley, dry bean, bred as inbred purelines, alfalfa, open pollinated populations are synthetics, hybrids, sugar beet and corn, clonally propagated varieties, we grow some potatoes. My own breeding program, Laramie Medic and Wyo Winter Pea, which is just coming along right now, are releases that fit into the IPL category. Lander alfalfa that we released to address a particular disease problem in Wyoming would fit into the open pollinated population or synthetic a somewhat more refined type of variety. Well, what are the modern tools? Whoa, that's not a complete list. Starting up here at the upper left-hand corner and over into the top here on the right side, I guess all of that fits into conventional plant breeding. Some of this is actually fairly new. We manipulate chromosomes, bits of chromosomes, whole genomes. And then there's a long list of in vitro tissue culture techniques. That's one of the areas of biotechnology, as I had them illustrated earlier. The molecular marker-assisted breeding, I'm not going to do justice to here, but genomics and now whole genome sequencing and so-called genomic selection fit into that. And then the genetic engineering or transgenesis, moving genes from one organism to another via recombinant DNA technology. And we have something coming along called cisgenesis, where we're maybe taking a gene out of a particular plant species, tinkering with it, and then putting it back. And is that GE or GM? My good colleague Sadnan Bekni up at Sheridan is doing some work like this with grape. And he asked USDA regarding cisgenesis and the types of regulations that might need to be followed and whether or not this was genetic engineering. And I think USDA told him, we'll get back to you on that. Well, obviously we don't have a time to discuss all of those methods and technologies. I am going to give you 8 examples for breeding and for biotechnology. Again, I'm not here to refute that speaker at the conference a few weeks ago. What I'm trying, I think Andrew did a pretty good job of refuting a lot of that stuff a couple of weeks ago. I'm here to put genetic engineering in the context of all the other things we do in terms of genetic modification. And maybe I've got more up there than I have time for, but we'll see. I'll be watching the clock. We'll get going. First a question. How many genes does it take to make a plant, people? Does it help if I tell you that it's about the same number of genes as it takes to make a person? No? I think it's about 25,000. About 25,000 loci, we would say, in genetics, places on chromosomes. And of course, we all get 2 copies of each, right? One from mom and one from dad, and it's the same way with diploid plants, and then multiply 25,000 times however many sets of chromosomes polyploid plants carry. My point here is that genetic engineering generally deals with one or very few genes at a time, whereas us plant breeders are messing with whole genomes. And in my opinion, I can imagine unwanted, unintended consequences from traditional plant breeding being more likely than from genetic engineering. I'll say that again towards the end. Well, emasculation. That's at the top of my list of techniques. To hybridize normally self-pollinated plants, usually to create new inbred pure lines. Mendel's success had to do with working with 7 traits in pea. We in our pea breeding program, by the way, work with these same traits. Most of them are involved in defining different market classes and types of peas. And Mendel figured out how to successfully hybridize this normally self-pollinated plant. And there's a figure from the text we use in genetics at the University of Wyoming. What are we seeing up here? For the female, a flower on the plant we're using as a female, we're removing the anthers so it doesn't self-pollinate. And then we're using a camel's hair brush here to transfer pollen to the stigma of our female parent. Truth be told, it's too late once the pea plant has exposed its flowers like that. It's already self-pollinated. The sexual column is down inside a pair of petals called the keel. It's really a flower in the bud stage, like near the top at the picture on the right, that we use as a female flower. And we will remove in the bud stage the sepals and the petals, and there we've got a nice stigma ready to receive pollen from the male parent. This doesn't occur in nature, emasculation like this. I originally learned emasculation working with soybean and with tiny flowers in tiny buds. I think we have another soybean emasculator in the room. Our new department head, Jim Heitholt, I believe. It's tough, isn't it, Jim? Yeah, it's tough. But anyway, I don't think there's anything terrible about this. I'm just saying it doesn't occur in nature. I don't mean that means it's unnatural. But doing this sort of thing, we've crossing different lines of pea. This is the one spot where I'm going to throw in just a little bit of my own research done with collaborators. We made lots of crosses between different pea lines and then advanced them at R&E centers in Wyoming, gradually reducing the number of lines we were maintaining, increasing seed of the best. F2 space plant nurseries, lots of hieroglyphics up here, F3 progeny rows, eventually getting to some pretty good stuff. An index of merit above or below the mean. All those 5 on the left are above. They are our products. The stuff off to the right, Common, Spectre, and Wyndham, C, S, and W, are existing cultivars. Pretty darn good cultivars, but bred in the Pacific Northwest and not so well adapted to here. Oh, our number 12 and our number 10 didn't do so great either. They were a couple of pets that we just couldn't let go of because they had some unique morphological anatomical traits. But plant breeding really, really works. And now we've crossed our most hardy winter peas, feed peas, with food peas. And we've got some food peas that'll survive Wyoming winters. And we think you could plant in your garden in the fall, and they would establish over winter and come on in the spring earlier than any seed you put in the ground. And in fact, this morning I was working with some middle school kids over at Levante Park at that community garden over there, getting them involved in plant breeding. And yeah, I'm for moving on all fronts. Participatory breeding, local breeding, amateur plant breeding. And by the way, regarding some other issues, organic, yeah, let's be as organic as we can be, using natural processes to control pests and pathogens, etc. But I'm not a purist. And by the way, organic farming systems are different from conventional, and people are breeding plants for organic systems nowadays. Back crossing. This is essentially gene transfer. We've been doing it for a long time in plant breeding. It may be intraspecific or interspecific, or involving plants from different genera or families sometimes. It may involve biotechnologies such as embryo rescue to make this sort of thing work. The following figure comes from a plant breeding text I used to use. And it's a bit complex, but what I'm labeling as the recurrent parent up there is a good agronomic or horticultural variety, okay? But it's got a problem. Some new pest or pathogen has entered our agroecosystem, or maybe a previously existing one is mutated in such a way that what was resistant is no longer resistant. So we go looking in related germplasm for resistance. And maybe we find a donor parent. In the textbook figure here, it's listed as a disease-resistant cultivar. Well, in fact, it may be a very inferior cultivar in terms of agronomy or horticulture. It may not even be a cultivated variety. It may be a wild relative, could be coming from a different species, could be coming from a different genus. Well, we cross the two. And if they're distantly related, maybe we use this biotechnology of embryo rescue. On the mother plant, the seed doesn't form, but we can extract an embryo and put it on an artificial medium in a test tube and grow up a plant. That doesn't occur in nature. And then we back cross it to over and over again to what we're calling our recurrent parent. And we're going to select for resistance throughout, eliminating susceptible plants. I think we have nice one-to-one segregations there, as per Mendel's first law, a couple of self-pollinations to get something that's true breeding resistant. Along the way, we're diluting out genes that come from the donor, approaching the genotype of the recurrent parent. But really, we're not just transferring generally that resistance gene. We're also transferring anything that's tightly linked on the same chromosome to our resistance gene. Maybe a more precise way of doing this is to genetically engineer that resistance gene from the donor into what would be our recurrent parent here, and we'd be less likely to have unwanted, unexpected results. Well, F1 hybrid cultivars, these are kind of a big deal in American agriculture and world agriculture. Maize, many other species, hybrid vigor. They don't breed true, which means farmers can't save seed without losing some of the hybrid vigor and without the seed produced on an F1 hybrid plant producing plants that segregate all over the place for traits like height and maturity. This is a great deal for the seed companies. They just keep you got your farmers coming back for seed every year. Yes, inbreed maize and yields go down. But it's possible to find pairs of inbred lines that, when combined, exhibit hybrid vigor. Like we're seeing here with whole plants and with ears of corn. And yield is going to be off scale. If you're going to, in the Midwest, in the Corn Belt, win a corn yield contest nowadays, you've probably got a field yielding well over 300 bushel per acre, and you are using hybrid corn. And it's because of all of this that we actually have about 2 million acres of corn fields in the U.S. that look like that, where the female parents have been detasseled, okay, and they're the plants from which you'll harvest your F1 hybrid seed, and that's to plant the 50 million acres or so of maize that we grow in America every year. Plant breeding really, really works. This figure was put together by a fellow named Troyer, who I believe was head of plant breeding at DeKalb for many years. Yields of open pollinated varieties are not that great. Some of our heirlooms aren't that great. There was an era called the double cross era that preceded the big single cross, and there's product in the middle here of a couple of inbred lines, good F1 hybrid, produced by a single cross. In the biotech era on the far right, we see even an accelerated improvement in yield beyond what was going on previously. And that steeper curve regarding the biotech GMO, it says up there, is not just due to the biotech traits. At the same time, improvement is still partially due to conventional plant breeding. I've squished this a little bit. Oh, and after after the Civil War, that's when our land-grant universities were founded, and people started collecting data like this. I've made room for 2014. What I read on the web yesterday is we're expecting yields of 164.7 bushels per acre, and a lot of the crop is in already, and that's probably a pretty good figure. There's some other interesting things contributing to this. One is creating inbred lines from haploids, where we cut the number of chromosomes in half. Some interesting tricks actually come up with by a company called AgReliant that I had an opportunity to visit near Indianapolis a couple of years ago. Their technique for extracting doubled haploids it's another revolution in the corn breeding business. Allopolyploidization. There's a mouthful. It's something that occurs in nature. My example here is going to be triticale. In fact, we see this particular mechanism of evolution occurring in the wild. Hey, has anybody ever told you, I don't believe in evolution because nobody ever saw a new species evolve? Yeah, via this mechanism we do. And we're doing it ourselves. See, we've got an intergeneric cross and subsequent chromosome doubling here. We're going to start out with bread wheat, which itself is an allopolyploid. Many sets of chromosomes, 6 here. 2 sets of A's, 2 sets of B's, 2 sets of D's, some of them tracing back to einkorn and emmer wheat and the dawn of agriculture and civilization. We refer to this as an allohexaploid, 6 sets of chromosomes. It's a lot of chromosomes there, 42. Well, not so long ago, really, in Northern Europe, Scandinavia, people were interested in growing small grains related to wheat that would have even more winter hardiness than bread wheat. And so they crossed bread wheat with rye, a nice diploid. 2n equals 14 chromosomes. There's our cross. And so they came up with something that's an interspecific hybrid with 4 sets of chromosomes, A, B, D, and R. These things are sterile, however, because chromosomes in the A set don't have any other A chromosomes to pair with, okay, and so on with the others. We get out a chemical that comes from the bulb of some kind of crocus or something, and I have no idea what its function is in life of the crocus. But colchicine is a chemical that interferes with spindle formation at mitosis. Some basic biology here. When that happens, chromosomes after mitosis end up in the same cell, and we double the chromosome number. It's sort of a chance event where you've treated growing points of growing plants. Maybe you'll see a branch that's bigger and more robust and fertile. Okay, and it's from there that you get your seed for your new allopolyploid, which in this case is triticale. The taxonomists refer to it this way, triticosecale, and it is fertile. We are dealing with whole sets of chromosomes here, and each chromosome has 25,000 genes. We're messing with a lot of genes in classical plant breeding. I think most of the anti-genetic engineering zealots are pretty much ignorant of some of the stuff we're actually getting away with here. An allooctoploid. Oh, and by the way, the plant brown-eyed Susan that gave rise to our University of Wyoming colors, I think they were used as centerpieces at an alumni function in the late 19th century. They were trying to decide what our school colors should be, and they had Rudbekia sitting in the middle of everybody's table, and they decided to go with brown and yellow. And there are some plant breeders right now reconstructing this species from its diploid progenitors to make better plants for ornamental and cut flower purposes, I believe. Micropropagation. This one I'm going to illustrate purely with pictures. African violet, regeneration from callus culture. For the most part, we're talking propagation here and not genetic modification. But the techniques used in micropropagation are a part of genetic engineering as well. So you got a violet you like. You go to its foliage, probably sterilize it, surface sterilize it, everything you're going to, and put it on an artificial medium, which allows plant cells to grow. All of this done under sterile conditions. We're looking at pieces of callus, disorganized cell growth. The magic ingredients here are growth hormones that you add to the medium, along with the mineral salts and sugar probably as a carbon and energy source. And then you change the growth regulators and put this under light, and you've got callus greening up and starting to regenerate little plantlets, which can go to Jiffy 7s, peat moss pellets, and eventually to that important scientific tool, the Dixie cup, and ultimately onto whole plants. I don't have a reference up here. I apologize for that. This was on a high school science website. A high school class did this and took these pictures, and I didn't save the reference, and I think that website is gone. But yeah, it was kids that did this. Oh, some interesting things do happen, though. I guess violet is fairly stable in cell culture, and you can regenerate plants that come out looking pretty much like what went in. But often there are mutations that occur, something called somoclonal variation, and it can do things like activate transposable elements, which explains that unusual soybean flower that we once had on the cover of the Journal of Heredity. Well, somatic hybridization. And there's more going on here. This one is in potato for resistance gene transfer. There's also sexual hybridization going on and back crossing and aided with molecular markers. This story comes out of one of the texts we've used in plant breeding. This has to do with virus resistance. There's a species of potato relative called E. tuberosum, which has resistance to a particularly nasty virus in potato. And to get that resistance into potato, these workers cut the number of potato chromosomes in half, crossed with another potato relative, and then took individual cells of these hybrids and the resistance source, and in a liquid culture stripped off the cell walls, got the protoplast to fuse, and were able to regenerate plants that they could then back cross to potato. There's a whole lot of stuff going on there that doesn't occur in nature, but it's pretty darn useful for humanity. And we're messing with those sets of 25,000 genes. I don't know that there's any varieties released from this particular project yet. Oh yeah, there's some back crossing. Geez, it's a couple of back crosses to potato, and you're back to something that looks like potatoes. Cell selection. Here's an example in corn for herbicide resistance, actually preceding glyphosate resistance and Roundup Ready, and it involves mutations that occur in culture, so-called somoclonal variation. Emi corn. I think the pioneer people, at least, sold quite a bit of this stuff at one time. It got onto the market quicker than Roundup Ready did. Am I right, weed scientists? It's kind of disappeared now, perhaps. But at a small biotech company in Minneapolis, I think, some workers started out with Mexican black sweet corn because it's something you could regenerate from culture in those days. I think they've got better techniques for regeneration of plants from cells in culture. And the mature corn is black. It's not black when you eat it, like on the right up there. Well, you take your explants and get them into culture, undifferentiated tissue, callus growing in culture. You throw in your imidazolinone herbicide. I barely was able to say it once, and I won't be able to say it again today, but I forget which one, but it's an ALS inhibitor, right, weed scientists? And you culture and reculture in the presence of the herbicide. You kill most of the cells, but due to somoclonal variation, the site at which the ALS inhibitor affects the enzyme and whatever pathway it is, mutates, and cells keep growing, and you can regenerate plants, get them to plantlets and onto whole plants, and then you've got IMI-tolerant resistant Mexican black sweet corn, which is not exactly what the corn growers in the Corn Belt are looking for. So you do some back crossing, and you get your IMI tolerance resistance into good inbreds, and you make hybrids. Well, this was quite successful, didn't create any serious problems as far as I know, didn't work as well as some things we're doing now. There's a bunch of stuff going on there that doesn't occur in nature. Well, finally, genetic engineering, or GMO. I'm trying to convince you we should call these things GE plants. Everything is genetically modified. We're going to use a bacterium called Agrobacterium tumefaciens, which causes a disease called crown gall, and it inserts some of its own genes via a plasmid into plant cells. We've disarmed the genes. I haven't. The gene jockeys have disarmed the genes that cause the disease. And we're dealing with individual genes pretty much. So linkage isn't a problem versus it can be with backcrossing. By the way, these darn genetic engineers make more work for us traditional plant breeders. We're maybe doing some pre-breeding and post-breeding. Well, it's Halloween. A couple of weeks ago, Andrew got to genetically engineer banana. And today we're going to genetically engineer a little pumpkin. Punky the Pumpkin came with me today. By the way, the needle on here is one of those blunt end ones that you use for refilling ink cartridges so nobody gets hurt. The anti-GMO people, how many pictures like this are there on the web of tomatoes and apples and Lord knows what? I'm going to black things out here. Okay, we're having some fun with this. On the serious side, yes, we have to be careful with genetic engineering and probably with all of genetic modification as to how we deploy resistance genes and so on in the environment. So, okay, we're having a little bit of fun with this, but yes, there's a serious side. Really what goes on very typically is getting out your paper punch with some surface sterilized leaves of a plant and putting these on artificial medium, on a petri dish, adding in the TI agrobacterium, disarmed so it doesn't cause disease. You've got your transgene on that plasmid, and eventually you'll get some transformed cells, and you can go back to whole plants, and this is how most of genetic engineering goes nowadays, and versus via embryos, plantlets, and regenerates. Gee, we made it through all 8. Is genetic engineering successful in the United States? Well, almost all the soybean we grow is Roundup Ready, glyphosate tolerant. Cotton, we've got both herbicide tolerance and insect resistance. The same with corn. And really, when you put the herbicide resistance and the insect resistance together with corn, you're up over 90%, really. Is genetic engineering successful worldwide? Yeah. This figure only takes us through 2009. As of today, worldwide, we have grown 4 billion acre years of GE crops without any real disasters that I'm aware of. A little bit of weed resistance to herbicides out there, but that occurs with non-GE weeds as well. People mention superweeds to me, and my usual response is, name one. And they can't. They don't exist. There's no weed out there that Steve and Andrew can't kill one way or another. And I've never, whose joke was it? Never seen a weed that wasn't, that's hole resistant. And in Wyoming, I can't tell you with alfalfa. I know there's some GE alfalfa being planted for forage. It's greater than 0%. 100% of sugar beet in Wyoming is Roundup Ready, except for a few oddball plants that Andrew keeps around for whatever purpose. Most of our corn, GE. Some crops we don't have genetically engineered varieties yet. Oh, and then we also produce a lot of alfalfa seed in Wyoming, both conventional and Roundup Ready. Some farmers actually want the ability to be able to kill alfalfa with Roundup. It's how they manage their rotations. So far, as I know, GE and conventional are peacefully coexisting in our state. Is this stuff safe? Steve Herbert put this slide together. The Center for Disease Control and Prevention, over this particular period of time, well, every year, they record millions of illnesses due to foodborne problems, thousands of hospitalizations and hundreds of deaths. So far, no one has documented a single illness, hospitalization, or death due to consumption of GE crops. Is this stuff safe? Here's that NAS report of several years ago, and they had several genetic engineering technologies scattered amongst other conventional technologies. The bialistic transfer with a gun has pretty much disappeared. Most of the transfer is done with agrobacterium. They've got towards the bottom as one of them, and they're guessing. This really isn't based on data. It's just based on basic knowledge of biology. What they considered to be the most likely to give unintended compositional changes was mutation breeding. And they don't even have somatic hybridization and all that wacky stuff up here. Most of our GE crops are engineered via agrobacterium. There's something called Clearfield wheat out there that is herbicide tolerant. It was produced via mutation breeding, but that's considered conventional breeding. Wasn't hard to release that stuff. Oh, and we see stuff like this. There's a retired physicist who likes to put together correlations. Glyphosate causes autism, and I don't mean to make light of autism. It's serious. Other people will tell you it's vaccination that causes autism. So use of glyphosate and autism both up. I think maybe autism is just more likely to be diagnosed nowadays. A fun group called the Literacy Project found an even higher correlation of autism with organic food. Now we're getting further. Here's one Andrew used a couple of weeks ago. This was an illustration in a journal that retracted the paper. They're back on a website, open source journal, that I guess states explicitly that your results need not be conclusive, and apparently you don't need controls. Here's the hardest question I'm going to answer, kind of finishing up. I want people to consider whether GE is in fact safer than some of the conventional things we do. Manipulating small numbers of genes rather precisely compared to messing with all 25,000 loci in plants. Transgenic is more specific now. Good grief. All right, 4 billion acre years of these things. Where are all the catastrophes, the catastrophizers have catastrophized? And I'm giving the short shrift, but you know, people plowing is not a natural process. We're saving a lot of soil, conserving water with GE crops, etc. How many genes does it take to make a glyphosate-tolerant sugar beet plant? 25,000 plus 1. Pretty specific what it is we're doing here. Yes, and we have many modern tools. How many of them are heavily regulated? Just that one. Who regulates these things? Oh, we got all sorts of agencies involved. Who regulates all this other stuff we do? Pretty much nobody. Maybe the marketplace to some extent. Is conventional breeding always safe? I don't have time to tell you the celery sunburn story in detail. Breed for insect resistance in celery, and it produces a chemical that kills insects but renders your tanning pigments useless. We do have something called generally regarded as safe. Some things are measured for toxin and nutrient content that do come out of classical breeding. Is concentration in the seed industry good or bad? I don't know. I mean, I promote moving ahead on all fronts. Yeah, and let's keep some of these heirlooms around. But this really, a lot of people find this really upsetting that several big companies have bought up so many little seed companies. There's still a lot of competition, though. Compare that to when I was a kid and there were only 3 automakers around, kind of keeping American motors alive, making ramblers so they wouldn't get hit with antitrust and monopoly stuff. A friend of mine asked me, do you want jellyfish protein? He told me he didn't want jellyfish protein in his corn. Do you want it in your corn? Probably not. Probably not the corn you consume, but you can learn a lot with fluorescent proteins from jellyfish. Our own Ann Sylvester was part of a review paper using fluorescent proteins to study basic cell structure in maize, which can only help us out in the long run. How about producing pharmaceuticals in plants? Maybe not in food plants. Boy, people don't seem to complain about genetically engineered microbes that make insulin and human growth factor. Is food being held to a double standard? What's the legal definition of a GMO? Well, we don't have one in America. Europeans talk about natural conditions, natural recombination. I just showed you 8 different techniques, or 7 besides genetic engineering, that don't occur in nature. Everything we looked at was genetic modification. Is every seed you plant a GMO? Well, that distinguished journal, the Farmer's Almanac, this lady gets it. She's actually promoting heirlooms and organics. But, yeah, that heirloom tomato and those seeds from her garden are GMO. All food, all plant foods are genetically modified. I've gone over. I apologize for that. Thank you.